It's a great pleasure to welcome Emmanuel Procyk, who has apparently been with us since 2002. So time goes quickly when you're having fun. Emmanuel is one of the heroes who still do single unit recording in behaving monkeys, and it's a very, very hard task. But if you're going to understand something about the cellular mechanisms of consciousness, decision-making, or what have you, at the end of the day, that's what you've got to do. And so it's a great pleasure to give the microphone to Emmanuel.
Thanks, Henry. Thanks everybody. Happy New Year. So today I will try to present some of the work we're doing in the team Neurobiology of Executive Functions. Here you have the list of the members of this team, and here their picture. I will not talk in detail about all the projects we are running, but basically we are all interested in how neural networks, and in particular those connected to the frontal cortex, are involved in organizing complex and flexible behaviors. So today I will focus on the cingulate cortex, but in a few weeks Charlie Wilson will speak about other aspects of prefrontal function. So my talk is about the cingulate cortex, but it's maybe more fundamentally about maps, cortical maps, and how they are organized and how we can study them precisely. And it's about primates, and in particular how non-human primate studies have been very important to instruct cortical function and organization in humans. So the cingulate cortex is represented here in the two species, in macaque and humans. It's this orange area here, which is surrounding the corpus callosum, and it's limited dorsally by a big sulcus, which is called the cingulate sulcus, here in the human and here in the monkey. It's a big piece of cortex, and it has been very difficult to understand, and there is still a lot of debate to understand what is this cortex doing for behavior and how it's functionally organized. And I think I will try to give you some insight about this. So we'll start with a bit of background and history about the function of the cingulate. Basically, the history starts with the circuit of Papez and the limbic system, which the cingulum is part of, and its function in emotional reaction, emotional processing, and behavioral control. And the cingulate has also it's a very interesting actor in the history of psychosurgery. In the 30s, 50s, and 40s, several investigators like Fulton, Pribram, and Jacobsen, for the most famous, started to investigate what specific lesions and stimulation of the cingulate cortex in both human and monkeys would induce in this species. And they observed that lesions and simulations were evoking autonomic responses, loss of tension, reduced reaction to frustration, apathy, and other dysregulation of emotional reaction and control on behavior. So that led this author to try to design a specific technique of lesion, in particular for humans, to treat and to create treatment for several psychiatric disorders. And this is an example given by Scoville. It's an undercutting of medial cingulate gyrus. And you see this at that time in the 50s, this was a very big and large lesion coming from the frontal lobe, the frontal pole, and removing the entire part of the rostral cingulate cortex, the gyrus, and the medial frontal cortex. This was highly unselective lesions, and indeed it was used for several psychiatric disorders like addiction, assortive behavior, schizophrenia, anxiety disorder, intractable pain, and above all, major depression and obsessive-compulsive disorders. So it's interesting that even today in some countries, in particular in the United States, we're still using cingulotomy, in particular to try and treat people with resistive major depression and obsessive-compulsive disorder resistant to behavioral therapy or pharmacological therapy. Here's an example of what people are doing these days. Here's an example from 2004. This is electrocoagulation of this medial part of the cingulate cortex, and that's for obsessive compulsive disorder. So it's stereotaxically targeted. It's rather small. It's based also on MRI. But to tell you the truth, it's not really clear why they actually decided to touch this lesion, this part of the cingulate cortex. And I don't think there is real data to support these targets. So anyway, this region here is exactly the region I will be interested in today. It's called the mid-cingulate cortex. And that's just a slide to show you the nomenclature. It's based on the, I think, the best subdivision framework put by Vogt with a neuroanatomist. It's based on cytoarchitecture and mapping of receptors in the cingulate region. So this is the mid-cingulate cortex, and what people call the anterior cingulate cortex, in fact, in this nomenclature is here, this rostral part which is surrounding the head or the genu of the corpus callosum. And I propose, it would be really nice if everybody would agree to use this sort of nice nomenclature. So what is the MCC doing? Well, in fact, if you look at the literature, in particular in humans using brain imaging techniques, there is thousands of papers showing activation in this region for a wide variety of tasks and different contexts. And so it's very difficult to come up with something really conclusive about the function of this. And one way to do this is to use meta-analysis. And I choose two, which are very nice one from Perron and Garcia-Larrea in 2000, and the one from Picard and Strick. And I took some of their data to map on this schema of the mid-cingulate cortex. So here is represented regions where studies have found activation in this region. So it's clouds of points from several studies. And it's related to different domains in which it was tested with different tasks. Emotion and affect in blue, pain in orange, attention in light green, and cognitive, like cognitive control or conflict detection in red, in dark green, sorry. So you see that the sort of overlap, you feel like there is some kind of specificity, but it's really not very clear at that point what, if we can subdivide this region in functional entities. And to add on top of this confusion, I have to talk about cingulate motor areas. Cingulate motor areas have been discovered and defined in the monkey, and these are cortical zones in the cingulate that project directly to the spinal cord and to the primary motor cortex. In the monkey, there are 3 of this region, and they have body representation. You have representation for the lower limb, the arm, the face, and they are separated in this cingulate zone. Here, by doing this meta-analysis, Picard and Strick more or less recovered 3 regions, cingulate zones, and you see that 2 of them actually overlap with this mid-cingulate cortex and the region involved in attention, cognition, or pain processing. So what do we do with this? Well, it's been very difficult, and actually if you look at the literature, you can find a long list of theories about what this region is doing, going from error detection, pain processing, conflict detection, cognitive control, emotion, attention to action. There are some link sometimes between them, but it's really unclear if we can make a conclusion out of this list. So I will argue that part of the problem here comes from the way studies are performed, and in particular a problem with brain imaging averaging. But before going into this, I want to show the contribution of studies in monkeys, and in particular single unit recordings. So this is a monkey brain, the medial surface, and you see the cingulate sulcus, which is open here in light brown. And inside, there is a representation of the cingulate motor areas for the arm representation, which is the rostral cingulate, dorsal and ventral, the 3 cingulate motor areas. Most of the recordings that I will show you that we perform and the majority of recordings that are published in the literature have been made in this dorsal part here of in the dorsal bank of the cingulate sulcus, encompassing some part of the cingulate motor area, the rostral one, and the region anterior to it, and that would correspond basically to this zone here in the human brain. So what I will show is that in these regions, neurons reflect that the mid-cingulate cortex is involved in monitoring behavior under uncertainty in situations where the monkey needs to adapt to its environment. And related to this, it's involved also in monitoring outcomes that are, of course, fundamental information to adapt behavior, and information that is used to evaluate action and update values to make appropriate decisions. So these are all functions related to adaptation and which relate to the cingulate cortex. So monitoring behavior under uncertainty. So that's the study we did with Jean-Paul Joseph. And the idea was to train monkeys in a task in which they had to find by trial and error which sequence of three touches, three movements on the touch screen they had to perform to obtain a reward. Okay, so there were 6 sequences, and the monkey had to explore all these sequences touch by touch to find by trial and error which one was correct, which one led to the reward, and as soon as the monkey discovered the correct sequence, he could repeat it and exploit his discovery to take more and more rewards. So we recorded in this cingulate, and what we found is that the majority of neurons that were active during the execution of the sequence, these are for different trials, these neurons showed a strong activation around movement, right? Before, during, and after movements, during all the period where the monkey was more uncertain about whether or not the sequence was correct and whether or not he would get his reward at the end. In this task, by trial and error, the monkey could actually, after some trials, predict that the sequence he had found would be correct, even before getting the reward. And in this case, we show that neurons completely stop discharging. And the majority of these neurons, most of these neurons, stop discharging, even if the monkey didn't get the reward, but he had predicted that this was correct, and this activity stayed low during the exploitation period. So mostly active during the uncertain condition. Later, Stan Dehen and his group, using the same protocol in humans, show by fMRI that indeed, when you compare activity during exploration, either search to exploitation routine, you have a strong activation of the mid-cingulate cortex with the striatum and other network involving the parietal and the lateral prefrontal cortex. So during this uncertainty period, monkeys adapt. And for adaptation, one important aspect is monitoring outcomes, and in particular, negative outcomes. And there is a long history, we heard a talk about this morning, history of the role of the mid-cingulate cortex in processing negative outcomes. It started, again, a bit of history, by monkey studies by Brooks and Gemba in '86, showing that by recording potential close to the cingulate cortex, you could record negative deflection for incorrect movement performed by the monkey during learning compared to correct performance during this task. Later, of course, in humans using EEG in the 90s, there was this discovery of what we call the error-related negativity. It's a negative potential that appears when subjects perform incorrect responses or start incorrect responses. It's a negative deflection compared to correct decisions and correct movement. This ERN is represented here on the surface reconstruction, and if you look at the source and try to reconstruct the source, most of the reconstruction lead to the mid-cingulate cortex. Later, actually about 10 years later, actually was discovered what we call also the FRN, which is a feedback-related negativity, which is a negativity that in response to a feedback, a sensory feedback given to the subject to give an output on his performance, whether correct or incorrect. And again, most of the reconstruction lead to a focus or major focus in the mid-cingulate cortex. We showed in monkeys that using ECOG, surface ECOG, in this exploration task, we can evidence a similar feedback-related potential in the midline. And when we record neurons, single unit activity in the cingulate cortex region I was talking about, it's very easy to find neurons which activity is selective for negative feedback, which is here the absence of reward compared of correct or presence of reward. Okay, so that all these studies led to this idea that the mid-cingulate cortex is involved in processing error. This is the error detection theory, or the negative affect theory. However, in our research, we pursued this experiment on this explore task. And in fact, what we found is that it's not selective to negative outcome. It's more responding to any and monitoring any outcome that is relevant for adaptation. So is, again, it's the same type of protocol, trial and error. The monkey here is exploring 4 targets. Only one of these targets is leading to a reward. So the monkey explores by trial and error. When he gets when he discovers the correct target, he gets its first reward. And after this first reward, he can repeat. And that's what we call the exploitation period, and in which he can obtain certain reward and accumulate these rewards. We did recordings in the cingulate cortex and first analyze the local field potential, and we show on high gamma oscillations, so this is the type of oscillation that Jean-Philippe Lachaud described to you a few weeks ago. This high gamma oscillation in response to feedback were mostly present for feedback during the exploration, whether incorrect or correct. And these were much stronger than the activity you can find during feedback, positive feedback during the exploitation period. And if you look at single neurons, the specificity of neurons responding at this feedback, well, what you find is that there are several populations and neurons that are obviously selective to negative feedback, here in red, and other populations that are selective to the first correct feedback, the first reward, that actually signal that the response was correct, and that signals also the transition between the exploration and the exploitation period. And during the exploitation, there is no response to the reward anymore. So this type of result and other, I will not enter into detail, led us to conclude that this mid-cingulate cortex is involved in discriminating any feedback that is relevant for adaptation and possibly for different types of adaptations. Last point, I will go quickly about it because I don't have the time to do it. It's about valuation of actions. Of course, outcomes, positive and negative, can be used to update the values for each decision and then try to decide the most appropriately. But what we show, and that was the key paper by Céline Amiez in 2006, in which we show that actually neurons in the cingulate cortex would code the expected average value that the monkey could obtain or expect during a task. This has been shown later in other papers, and actually some groups like Dale Lee's group showed that also you could find traces of the reward history, the history of the reward or outcomes that the monkey get in a succession of trials that was encoded in the cingulate cortex. And more recently, the group of Matthew Rushworth showed that this anterior mid-cingulate cortex was actually as an activation that correlates with the value of the environment to explore, the value of the exploration compared to the value of certain straightforward decisions. So if I want to conclude on all this data and make a package out of it, I will use this title of this nice paper from Tim Berenson and Matt Rushworth, who showed that the mid-cingulate cortex is involved in learning the value of information in an uncertain world. And I think some of the data I just presented to you goes and fits directly into this statement. So okay, that mostly what monkey studies brought to us regarding MCC function. Does that fit with this, all these theories? Well, not exactly, and in fact there are many disagreement in the community, in particular between monkey and human studies. And this led some authors to suggest that actually this divergence of data between humans and monkeys played for a real difference in nature between the mid-cingulate cortex in humans and the mid-cingulate cortex in monkeys, saying that during evolution, the mid-cingulate cortex in human evolved differently and created a new area which is not present in monkey. What I want to show is actually the reverse, and I think we have all the data to say that there are convergence between data obtained in humans and monkeys regarding the function of the mid-cingulate cortex. That's what I will show now, and it's mostly based on the recent data obtained by Céline Amiez in functional brain imaging, and it's about the subdivision, the motor maps, and the cognitive function in the mid-cingulate cortex. Okay, why do we have a problem with brain imaging studies? I think part of it is related to the fact that the main approach is to average people, average brain, and not taking into account their morphology. And in fact, it is known, and it's very true for the mid-cingulate cortex, that there is a very strong inter-individual morphological variability in this region. Here is a typical example. In all of the brain, what you have is what I showed you earlier. It's a cingulate sulcus, a long sulcus that goes here in this direction. But in 75% of hemisphere, you have a secondary sulcus, which is shown here, which is called the paracingulate sulcus. It's just above the other one, and it extends from the anterior cingulate cortex to the caudal part of the mid-cingulate cortex. Okay? It's 75% of hemisphere. Sometimes you have one on the left or on the right, or you have two, or you have none, depending on the subject. What Vogt, the anatomist, showed is that the presence and the morphology of this sulci actually impact on the disposition of cytoarchitectonic areas. So he did the cytoarchitecture in humans, and he showed that when you have only one cingulate sulcus, area 24, the subdivision of area 24 up to area 24C, lies in the gyrus and the ventral bank of the cingulate sulcus. And above it, there is an area 32 prime, which is in the dorsal bank of the cingulate sulcus. When you have a paracingulate sulcus, where area 24C actually goes up in the cingulate sulcus up to the dorsal bank, and area 32 prime goes up in the gyrus above it and in the ventral bank of the paracingulate sulcus. So if you would average this brain, actually you would mix up areas that are from different cytoarchitectonic type and possibly from different functional type. So what do we do with this? Well, what we can do is single subject analysis, and that's what Céline Amiez and Michael Petrude started some years ago, working on the frontal eye field in particular, and they applied this to the study of the cingulate motor areas and tried to relate this with the morphology of single subject. So this is fMRI. It's a simple protocol in which you ask subject in blocks to do a simple movement of the eye, of the tongue, rotating the tongue, repetitive movement of the hand or of the foot, okay? And you look at the peak activation for each of these movement. And what they show here is a representation of the average peak for the population. You see, and the peak taken subject by subject, you see that they could reconstruct 3 cingulate motor areas below the SMA here, okay, which lies in the cingulate sulcus. And you see that the 2 more rostral cingulate area have representation for the lower limb, the arm, the face, and the tongue and the eye, okay? This is like in monkeys. In monkeys, we have 3 cingulate motor areas. These are for the arm, but actually we have also leg and face representations. But the most striking is that if you look at subject with a paracingulate sulcus, what you observe is still these 3 cingulate motor areas, but the face representation is going up into the paracingulate sulcus systematically, okay? So that shows that there is an interaction between how representation are disposed in the brain and the single morphology of each subject. how does that speak to the cognitive function of the cingulate, in particular its role in feedback-related activity? So what I showed you initially, that we design in monkeys task, the trial and error or explore task, which is very powerful to discover the properties, the functional properties of the mid-cingulate cortex. Where we use this task in humans, it's adapted to humans, but we kept in this study one major point, which is in monkeys usually we use juice as a reward. In humans we usually use other things like money or abstract feedback, but here we use juice to be as close as possible to our protocol in monkeys. So subjects were deprived of water from the previous day. They were scanned in the morning, and they were doing this task in which they had to find by trial and error which of the 3 targets would be associated to the largest reward. Okay, so there are several conditions, I won't enter into detail, but basically on each block of trial, one is giving more juice than the 2 other ones. So the subject explore, sorry, the subject explore, and when he finds the target with the largest juice reward, he can exploit this juice until the next problem. So when we look at the activity at the feedback, and we do the average across subject, we find what we should find, which is an activity in the mid-cingulate cortex, that's for left and right hemispheres, that are stronger for feedback in the exploration period, incorrect and first correct, compared to repetition or exploitation. And if you look at the percent bold signal change in these different trials, the negative one, the discovery, the first positive one, and the repetition, you see this pattern of strong activation during exploration and disappearance of activation during the exploitation, like neuronal activity I described to you in the mid-cingulate cortex of monkeys. So, but the nice part is that the location of feedback-related activity in the mid-cingulate cortex is actually predicted by local morphology. And here I give you 2 single examples. When in this hemisphere, when you have a cingulate sulcus and a paracingulate sulcus, the feedback activation, that is the activation which is stronger in exploration compared to exploitation, is in the paracingulate sulcus. When there is no paracingulate sulcus, the activation is in the cingulate sulcus. This is actually one single individual, and you can see on the frontal section the separation of for the left and right going in the paracingulate and the cingulate when there is no paracingulate. Another example here of a subject that has a reverse pattern. Here, no paracingulate, the activation is in the cingulate. When there is a paracingulate in the other hemisphere, the activation is in the paracingulate. Again, the coronal section. And if we look at all the subjects, you see that for the peak of activation, they are all in the cingulate sulcus when there is only this sulcus, and it's actually at the intersection with a vertical branch of this sulcus in red. And when you have a paracingulate sulcus, so that's all the sulci overlap for all subjects, then the activity is in the paracingulate sulcus, also contingent with this vertical branch. So there is a strong relationship between the morphology of subject and the location of this functional area, which is the area involved in feedback during exploitation, juice feedback during exploration. Okay, let's summarize this. We showed that the feedback activity is located depending on the morphology of each subject, and it's in the paracingulate sulcus when it's present, and in the cingulate sulcus when it is not present. Remember, Vogt showed that in this case, area 32 prime is here in the dorsal bank of the cingulate sulcus, but when there is a paracingulate sulcus, the area is actually in the paracingulate sulcus. So that's a strong suggestion, of course not a proof, but it's a strong suggestion that area 32 prime is actually the one to process juice feedback information during exploration. And that fits with monkey studies, in fact, because in monkeys I showed you that we were recording mostly feedback-related activity, which were juice feedback, in the dorsal bank of the cingulate sulcus. And in this dorsal bank, Petrides and Pandya and others show that there is a transitional area which is, could be similar to area 32 prime, although not everybody agree on this. Last thing, not last thing, I mean another thing, remember the cingulate motor area. Céline and Michael showed that the face representation is actually shifting to the paracingulate sulcus when there is a paracingulate sulcus. So possibly, and this face area represent eye movement and tongue movement. So there is a possibility that actually this juice-related information, which is actually taken by the mouse, correspond to a cingulate motor area corresponding to the face. Here we overlapped the single point study for the feedback in the study I just described with the, so that's the square, the orange square, with the region where all the peaks were found for the tongue area in the Amiez and Petrides study, and you see they completely overlap. Of course, it's two different studies, two different group of subjects, but there is a quite good overlap, and it suggests that feedback-related activity in the mid-cingulate cortex is processed or overlap with this sensory motor representation. So we tested this recently. We did the experiment in collaboration with Michael Petrides, and Céline went to Montreal for this, to do in the same subject the mapping for the tongue and the feedback-related activation for juice in the explore task. And these are the conjunction map for the different type of subject, and you see that the conjunctions between feedbacks and tongue movement or tongue representation is in the cingulate sulcus when there is no paracingulate sulcus, and shifts in the paracingulate sulcus when there is one. So that shows that the two maps goes together and are activated together, and probably feedback is processed by this face representation. I have to mention, I don't want to do it, but I have to mention that we control that it's not a problem of movement of the tongue, because the activation of the motor area related to tongue movement doesn't show differences between exploration and exploitation. So it's really the process of feedback during exploration. And then you can make another prediction. You can make the prediction that if the cingulate motor maps are involved in processing feedbacks, you could predict that if you use a tactile feedback on the hand representation, then according to the Amiez study, if you do this kind of conjunction map, you would have activity in the cingulate sulcus with a conjunction with the arm representation, and that would not shift in case of paracingulate sulcus, because the arm representation doesn't shift, it stays in the cingulate sulcus. But that's what we did, and actually the preliminary result show that it seems to be true. There is a conjunction map here in the cingulate sulcus that doesn't shift, so that's air puff on the hand as a feedback, and hand movement motor area, which doesn't shift when you have a paracingulate sulcus in the subjects. Okay, if I want to make a sort of conclusion, I don't have really a thoughtful conclusion about this. It's all work in progress, but we could say that the rostral cingulate motor area might serve as a body map-based monitor to process and detect behaviorally relevant information, relevant for adaptation. And a sort of embodied radar for adaptation, although I'm not sure this terminology is actually appropriate. But I think we can start with this. So to conclude this talk, I want to make a few comments, a moral about these studies. The first is that non-human primate brain and the study of these non-human primate brains is highly precious. It's a precious model to approach even complex human cognitive function and how human cortex is organized. The second comment, which is sort of a easy comment to make, the second comment is about mapping. And I hope you're convinced that to do a precise mapping of cortical function, actually we should really think to take into account the individual variability and the individual morphology of the brain. And doing the single subject approach, like what Céline is doing, allows several things which are very important. The first one I talk about is to do precise anatomofunctional definition and try to compare different conditions and how different subregions in the brain are actually processing the same kind of information. The second is to do, it's probably a requirement, to do a correct assessment of inter-individual variation in function. If you want to show that personality trait or psychiatric states correlate with particular activation in this region, you need to be sure that you are actually measuring the activity in the same functional region, and you're not just taking an ROI, which is average of a subject, because this ROI would actually shift from one subject to another. So single subject approach is very important in that respect. And last and not least, I think it would be important to use this type of approach, and I think that's the only way to do it probably, to instruct clinical targeting of subregion to remove or to stimulate in the cortex in particular, and that's actually what is done for deep brain stimulation, that recording, single individual recordings are used to really define the function of areas that are going to be the target of the treatment. Let's finish on questions for future research. There are tons of questions. I put three, which I think are very, we want really to address. The first is, what is the role of the cingulate to primary motor and spinal projection in feedback processing? These cingulate motor area actually project directly to systems that are involved in the production or the regulation of movement by themselves, but what are their role in feedback processing and in adaptation of behavior? How do cingulate feedback-related maps connect to large-scale networks? This is one project Céline started and is presenting to the CNRS, actually, and how these different subdivisions connect to a different area in the prefrontal cortex and in the rest of the brain. And finally, I think one of the most interesting questions is this. What are the relationships between feedback-related maps, the one I described, the body maps, and pain? Garcia showed us that the mid-cingulate cortex is involved in some aspect of pain processing. Is this pain processing actually computed by the same map, or are there different body maps for pain and for feedback-related activity and motor-related activity? I think that's really an experiment we need to do, and it's probably possible to do it within the context of cortex. I'm waiting now for your feedback. Thank you.
Questions on the floor? When you have the paracingulate gyrus, sulcus, you've got an expansion of area 24C, or the area is 24.
Well, there is a change in the morphology of the entire region, yeah. So the fact whether you have more cortex or less cortex is, I mean, there's some studies working on this. It's not really clear yet because it's sort of difficult to do. But it seems that there is a change in morphology, maybe in the depths of sulci also, that the cingulate sulcus would be maybe deeper when it's alone compared to when there is a paracingulate sulcus. But I think if we want really to go forward in that direction, we need more studies, in particular more cytoarchitecture in humans and more clear measurement of what this region is doing. Yeah, absolutely.
Questions from the floor? Stunned.
I was convincing, you know?
You were totally convincing, yes. Do you ever see this paracingulate sulcus in monkeys?
No, there is no paracingulate. So that's a very important point, actually. So there is no paracingulate sulcus in monkeys. And we, with Céline, we were thinking deeply to contact people working on great apes, because it would be interesting to see whether there is a sort of intermediate state in evolution in which you have appearance of this paracingulate cortex. Why it's interesting is because there's been a lot of correlation between the presence of the sulcus and some pathological state in humans, and that would be interesting to see whether it's actually there is something in lesser species like chimps or other apes. So we're going into that, but there is no paracingulate sulcus in monkeys.
Jean-René, I think, has got a question.
Yes. Manu, you showed some source reconstruction for EEG studies that look at what was it, error-related negativity. Is there any kind of direct evidence from monkeys or from human studies that this signal picked up at the skull level correspond to the signals that you record with microelectrodes in the cingulate cell?
There is no direct evidence, no. So in the monkey, there have been surface recordings. So what we did, and Jeff Scholl did some work on this too. There have been local field potential recordings in the cingulate sulcus. I'm not sure there have been, maybe recently, the SFN, there was some recordings of simultaneous local field potential and surface EEG that seems to go in the right direction, but there is no clear evidence. In humans,
so that supplementary eye field wasn't cingulate.
No, it did some recordings in the cingulate cortex too. Yeah, regarding errors in this saccade task. Like, so it's more an error-related negativity, in fact, in this case, because it's for the movement. It's not feedback. I don't know of any direct evidence. Actually, there is a possibility that this ERN or FRN has multiple sources. So maybe that the mid-cingulate cortex would be one main source, or maybe the closest source to the surface. And it's possible that, I don't know, the circuit involving the striatum or another deeper structure would be involved too. It's not really clear what is the real origin of the negativity.
Question over here.
When you showed the pictures of the different populations of neurons responding to either incorrect exploration or the first correct ones, I did not pay exactly attention. Did you mention that there were some neurons able to answer to both? Are there another lab between the two populations or not?
Well, I didn't mention it, but there is actually a small population of neurons that was also described by another group from Japan, the Tanaka group, which is not represented here. It's a group of neurons that are very fast responding and very short latency. They are very phasic. And so just at the appearance of the feedback, and that don't discriminate between positive and negative. But they are active for exploration compared to exploitation. So it's like, it's a very interesting question, in fact, because it's like probably there is a first wave of processing that doesn't discriminate between the stim, and within 150 milliseconds, then the areas start to discriminate and process the incoming information. So we sort of have both, right?
So, is the time gap between these two putative waves, would it be compatible with an exchange of information between two era?
No, it's too fast. It's really too fast.
Alexandre.
But, I think this is great stuff, and I believe this is one of the first clear examples of inter-individual differences linked to SALSI. I was wondering whether you know the human literature showing that the receptor distribution is actually different in similar-looking cortex on a gyral crown and in the depth of a sulcus. If that is so, and if you've got an example of inter-individual differences which are linked to something we can see with current MRI, namely SALSI, then you would, well, one might predict that there might be a difference in certain performances linked to that type of receptor. I'm thinking of NMDA containing 2B subunits, for example. Have you thought about looking at performance between subjects with the different morphologies that might be linked to receptor types in those areas?
Okay, so there are many points here. Important points. First of all, this is not the first study. Actually, Céline and Michael did the first real nice study about the frontal eye field, the relationship between the position of the frontal eye field and particular morphology of sulci in 2006 in General Science. And there are very few groups that goes in this line. But I think this type of study will expand quite quickly, and I hope will be the case. Regarding receptors, actually Vogt map the areas based both on the situ architecture and the repartition of different types of receptors. That's how actually he decided to separate different regions and different areas within these regions. I don't recall that he actually looked at the relationship between the morphology and the distribution of receptors. But he probably didn't, but we would have to look at the, these are papers by Palomero, Gallagher, and Vogt. You can find this. Okay, in our study, we checked that the behavior was the same or not between subjects. It was exactly the same. So we don't believe that actually these are different brains. On the contrary, we believe these are the same brains in terms of what they can do with information. It's just that the areas that are processing the information is just not in the same position in the brain. And that helps us to localize and look at the organization of these areas. But these are not, okay, the cingulate brain is less than the paracingulate brain. That's not the case as far as we are concerned. There is one study in neuroscience, Buda and colleague, who trying to go in this direction, showing sort of correlation between certain performance and the presence of the paracingulate sulcus. According to me, it's not very convincing, but I mean, this is an open question, and we certainly, I think the receptor idea is a very good idea. There have been some relationship between transporters also, dopaminergic transporter and performance. Was dopamine? I don't remember what they were targeting exactly, but dopamine transmission and feedback-related activation in the cingulate cortex, and they show. I think it was based on genotype. Maybe you know that, Jean-Claude. They showed that the activation of the cingulate, the mid-cingulate cortex, was different depending on the subject type, depending on dopamine transmission, and they were expressing different type of behavior. That some were more taking into account negative outcomes to learn, whereas the other one were taking more into account positive reinforcement. So there's definitely a field of studies to open here.
When you say that the presence of the paracingulate sulcus doesn't change anything, you mentioned earlier that there was a pathology associated with it, so that is a change.
There are maybe statistical indication. Well, I'm going to tell you one of the data which is in the literature, which is that there is an asymmetry. It seems that there is an asymmetry in humans. There is more, I think, more paracingulate in the right compared to the left. And this study I'm thinking about showed that this asymmetry is less pronounced or absent in a schizophrenic population. But it's an asymmetry of statistical presence of paracingulate cortex. So there is not a direct link between the pathology and the morphology. That's why I'm saying it's, there may be something, but it's not that clear.
Any other questions? Charlie? No? I thought you were waving your hand. Any other questions? Okay. Thank you very much, Manu.
Thank you.